Okay, so in the next two videos, we're going to be talking about the purine synthesis pathway. So we're going to begin by discussing the purine synthesis in more detail, and we're going to discuss the de novo purine synthesis pathway and the salvage purine synthesis pathway. And then we're going to move on to discuss purine degradation. And then we're going to move on to the clinical correlates of purine metabolism. So we're going to discuss the pathologies that arise due to abnormalities in the purine pathway, and we're also going to discuss drugs that target the purine synthesis pathway. The goal of the purine synthesis pathway is to synthesize the purine nucleotides that will be used in DNA and RNA synthesis. So the two purine nucleotides are going to be adenosine and guanosine. So there are two pathways for purine synthesis. The first is going to be the de novo purine synthesis pathway, and the second is going to be the salvage pathway. The first is called de novo purine synthesis because we make purine nucleotides from scratch. The second is called the salvage pathway because we recycle the nitrogenous bases and convert them back to their purine nucleotides. So we're going to begin by discussing the de novo purine synthesis pathway. So in this pathway, we're going to start with the ribose 5-phosphate, and then we're just basically going to keep adding molecules until we get the respective purine nucleotides. So until we form adenosine and guanosine. The first step in the de novo purine synthesis pathway is going to be the creation of the molecule PRPP, also known as 5-phosphoribosol 1-pyrophosphate. So like we said before, we're going to start with the ribose 5-phosphate and we're going to convert it to PRPP, and this is catalyzed by the enzyme PRPP synthase. So pretty much what we just did is that we took a pyrophosphate molecule and we added it onto the first carbon in the ribose sugar. The next step is going to be the creation of IMP, or inosine monophosphate. Now this is fairly simple. We just take PRPP and it's going to be converted through a series of enzymatic reactions to inosine monophosphate. IMP is actually one of the purine nucleotides, and its nitrogenous base is going to be hypoxanthine. Now the reason that we don't hear about it as much as AMP or GMP is because it's not used in DNA synthesis. The third and the final step in the de novo purine synthesis pathway is going to be the conversion of inosine monophosphate into either AMP or GMP. So the only thing that happens is that instead of hypoxanthine as a nitrogenous base here, we're going to have either adenine or guanine. So the second pathway for purine synthesis is going to be the salvage pathway. So what happens here is that our body salvages the excess nitrogenous bases that come from either our diet or from cell turnover, and it converts them back into their respective nucleotides. As part of the purine degradation process that goes on in our body, GMP will be converted into guanine, which is their nitrogenous base. IMP is going to be converted into hypoxanthine, which is its nitrogenous base, and AMP will be converted into adenine, its nitrogenous base. Now, eventually, all these nitrogenous bases will be converted into uric acid for excretion into the urine. The salvage pathway is composed of two enzymes, HGPRT, which stands for hypoxanthine guanine phosphoribosol transferase, and ARPT, which stands for adenine phosphoribosol transferase. So the HGPRT enzyme takes guanine and hypoxanthine nitrogenous bases and it converts them back into GMP and IMP respectively. The ARPT enzyme takes adenine and converts it back into AMP. So the following diagram depicts how the salvage pathway takes place in a bit more detail. So guanine and hypoxanthine nitrogenous bases are going to be added to PRPP by the enzyme 8-GPRT to form respectively IMP and GMP nucleotides. Adenine is also going to be added to PRPP by the enzyme APRT to form AMP. So just to recap the purine synthesis pathway, we said that we're going to start with the ribose 5-phosphate, and that's going to be converted into PRPP. PRPP is then going to be converted into inosine monophosphate, or IMP, through a series of enzymatic reactions. During that process, amino acids, aspartate, glycine, and glutamate are going to be consumed, tetrahydrofolate and CO2. So the amino acids are going to be the source of nitrogen, tetrahydrofolate and CO2 are going to be the source of carbons that make up basically the rings on the nitrogenous bases. IMP is then going to be converted into either AMP or GMP depending on the need. It's also important to understand how the purine synthesis pathway is regulated. So when we have elevated levels of IMP, AMP, and GMP, they are going to have negative feedback inhibition on an enzyme called glutamine PRPP aminotransferase. Now this is one of the enzymatic reactions in the purine synthesis pathway. So now we're going to discuss how purines are broken down inside the body. So we said before that basically we're going to start with the ribose 5-phosphate, we're going to form PRPP, and eventually basically we're going to get the three purine nucleotides. Now all these three purine nucleotides are going to undergo metabolism, and they're eventually going to form uric acid. So uric acid is going to be excreted in the kidneys and into the urine. So how does purine breakdown take place? So we're going to discuss here GMP and IMP because they have a very similar pathway for metabolism. So GMP is going to be converted into guanosine, and now this is going to be the nucleoside form. So basically we just remove the phosphate. Guanosine is then going to be converted into guanine, which is going to be the nitrogenous base. Guanine is going to be converted into xanthine, and then xanthine is going to be converted into uric acid by the enzyme xanthine oxidase. IMP is going to be converted into ionosine, so same thing, we just remove the phosphate. Ionosine is then going to be converted into hypoxanthine. Now remember, hypoxanthine is going to be the nitrogenous base. Hypoxanthine is going to be converted into xanthine by the enzyme xanthine oxidase, and xanthine is going to be converted into uric acid by also by xanthine oxidase. So AMP is going to be broken down into adenosine, and then adenosine is going to be broken down into inosine by the enzyme adenosine deaminase. Inosine is then going to be broken down into hypoxanthine, hypoxanthine to xanthine, and then xanthine to uric acid.